Good morning. It's my pleasure to be here at the 47th Annual Convention of the Philippine Society of Biochemistry and Molecular Biology. And I'm grateful to the organizers and the officers of the PSBMB for the invitation. I'm really quite excited also to share with you some of the progress that we've made on this research, which is wastewater based surveillance for SARS CoV 2. And before I start, I'd like to acknowledge our collaborators and our team from NC State University, Nadine Kotlers and Angela Harris. Nadine is a postdoc, and Angela is an assistant professor. And then we have collaborators from Rice University, Howard University, and USC. And also our collaborators across the state of North Carolina. And this is part of the Pathogen Research Network, and you can see some of the folks that are part of this network. So one of the things that is important in facing this current pandemic is determining the infection or the incidence of COVID-19 in a community. Basically, figuring out what's going on in a community in terms of infection. Is it decreasing? Is it increasing? Are our intervention measures working or not? And we can do this by testing individuals. But as you can imagine, this is costly. It involves a lot of tests, and if you have to do it over time, then it becomes even more expensive. One way of minimizing the costs is doing something called pooled testing, where you take a group of people and then you test that group of people, and that gives you an idea of what's going on in a community. But if you really think about it, there's really a much better way of getting a pooled sample from a whole community. And that is the first breakthrough in this research, is understanding that everybody poops. Everybody contributes to the wastewater. And if you're collected or connected to a wastewater treatment collection system, then really that's a logical and really nice way of collecting samples from everybody that's on the sewer system, because, like it or not, you are contributing to a sample. So this is called wastewater surveillance. And this is a schematic of the approach. You start with the household, and again, everybody in the household would use the toilet. And if you think about a neighborhood, a community, a whole city even, they're basically contributing to the wastewater. The wastewater is transported in sewer lines, and you may have all kinds of inputs. You may have other neighborhoods, you might have distal contributors. But at the end of the day, you're sampling the influent as it goes to the wastewater treatment plant, or maybe you're sampling the primary solids inside the wastewater treatment plant itself. If you collect those samples, and then you can do this again over time, take them to the lab for analysis, and then you can analyze essentially for the signature of the SARS-CoV-2 virus. And an important part of that also is reporting back to the stakeholders, the community, the city, the public health departments. This is called wastewater surveillance, and sometimes you'll hear the term wastewater-based epidemiology. And we use that term as a more general term if we are converting or relating that to the disease itself in terms of how it's spreading and so on. So I said that this was potentially more cost effective than clinical screening, and there is a recent publication that basically shows, at least in the US, 70% of the US population could be screened. There are about 15,000 wastewater treatment plants, and the cost for the reagents alone would be something like $225,000, which is not really a big number if you consider the amount of money being spent on individual testing. And that cost would really just be 0.014% of individual clinical testing. And you can do this again repeatedly. So this is the reason why, since March, wastewater surveillance for SARS-CoV-2 has become really a topic that a lot of research teams across the US and even globally are working on. The first report was end of March. This was in the Netherlands. And then in April, the first report from the US came out. And then obviously till today, in December, we still have a lot of groups working on this. So we're one of maybe 70 plus teams in the US trying to do this kind of work. So the very first question is, are we sure that wastewater is actually uh, are we actually really seeing SARS-CoV-2 in the fecal material? So this is a table from a March 2020 paper that basically shows that you can find SARS-CoV-2 in sputum, right? Basically, oral discharges, nasal swabs, pharyngeal swabs, and this is the common swab technique that's being used clinically, and also fecal material. You can see the feces is in here. So this is very consistent. It's accompanied by shedding in the stool. Anywhere from 27% to 89% of patients do this. And then you have a varying level of amount of RNA copies of the virus per gram of fecal material. And that varying level actually brings complications in terms of our estimations and quantification of the number of people in a community that's infected. But this is basically showing that it is being shed in fecal material, and that this is a sample where we can detect the virus. Now, one question that I've always been asked is that is this a way for COVID-19 to be transmitted? And the answer is that it is considered unlikely. SARS-CoV-2 is still spread through person-to-person respiratory methods and not by the fecal oral transmission pathway. The other thing to remember is that SARS-CoV-2 is actually very easy to inactivate compared to other viruses that may be in the environment or in wastewater. So if you look at this table, this is from the CDC. You have more resistant at the top to the most susceptible or easily inactivated at the bottom, and SARS-CoV-2 is at the very, very bottom. Even easier to inactivate compared to other viruses like HIV and hepatitis B, or some other bacteria that are infectious, or even some of the non-lipid or small viruses like the poliovirus or Coxsackie virus. So this infection is easily obtained or easily attained in SARS-CoV-2. So again, if you wash your hands, if you avoid crowds and being close to infected individuals, then essentially that's the best way to avoid getting COVID-19. So what do we mean by surveillance? We're really trying to quantify SARS-CoV-2 in wastewater. And we can do this in the influent, which is the wastewater coming into the plant, or we can do this through the solids, the biosolids, typically the primary solids. The very first stage of sedimentation produces these solids at the bottom where you can collect them and you can actually test for SARS-CoV-2 there. So these are the steps. You have sample collection, and then you might dose internal standards. And here I'm showing an example, a bovine coronavirus as our internal standard. You can store the samples, and then you can actually extract the RNA of the virus, and then perform reverse transcription, convert that to DNA, and then use different ways of PCR. Here I'm indicating digital droplet PCR to detect these targets, N1 and N2. And there are other targets that you can use that are specific for SARS-CoV-2. The cost estimate ranges from about $300 per sample down to maybe $70 per sample. It really depends on what you have already in place in terms of personnel and machinery. And you can get the results within a day, maybe more commonly 2 to 3 days. So what do we mean by detection? Really what we're doing is we're detecting specific RNA sequences in the virus, and this is showing the viral genome of SARS-CoV-2 and some of the targets. So there is a Chinese CDC target, which is the ORF1A, the WHO target, which is the E target, and then and you also have the CDC targets, the nucleocapsid proteins N1, N2, and N3. And to date, the most common targets are N1 and N2. So basically, this is what we're trying to detect. So that means that we are detecting the RNA and we are not detecting the intact virus. So we may detect RNA that's coming from inactivated virus, RNA that maybe just is naked RNA, just floating in the wastewater. And if we detect the RNA, it doesn't mean that we have infectious virus. And as I said before, there is really no evidence that there is infectious virus in wastewater to date. So that is an active area of research, but a lot of people are working on this, and the consensus is that we probably will not find infectious virus in wastewater. Um, the detection, like I mentioned, can be using qPCR, quantitative or real-time PCR. This is a particular method that I'm showing here is called digital droplet PCR or ddPCR, and it's a method that basically uses a different type of chemistry where you make droplets, about 10 to 20,000 droplets, and each droplet is where the PCR reaction happens. So the amplification happens in each particular droplet, and you have, let's say, 10,000 of these. And so each droplet will become positive or negative. And what you do to quantify is you basically count the number of positive droplets and divide it by the overall number of droplets. So if you detect 3 per 10,000 droplets, that's your detection. And that's your percentage. And so it's quite sensitive, and it appears to be the method of choice for this particular application. This machine, I'm showing you the Biorad QX200 ddPCR system. This machine is not inexpensive. It actually costs $105,000 for a basic setup. So, what are the potential uses of this type of data? The first one, and perhaps the most useful, is actually in terms of detecting trends. What are the changes over time happening in a community? Can we actually detect it ahead of the individual testing? So, for example, this current so-called third wave that's happening, can we detect it before it happens? We can also use it to look at the impact of public health interventions. If you have a quarantine QC of some sort, are you actually seeing the decrease in infection in a community, or is it not doing anything? The other thing that is important to a lot of researchers right now is the co-occurrence with other infections. So, we have flu that's expected to increase during the winter, and how is this going to, interact with the trends for COVID-19? Here's an example of some data from Valencia in Spain that shows that you can detect the RNA of SARS-CoV-2 about 2 weeks before you actually see the very first clinical cases. So wastewater here serves as an early indicator of what's going to happen in a community. So that's really interesting. Um, Here's another example, or another way that we can actually use this data, which is a little bit controversial, or at least questionable, because there are so many assumptions. And that is, how do we estimate the infection prevalence in a community? How many people are really infected in a particular city? And the advantage of wastewater testing is that even asymptomatic patients, the people who are not showing symptoms, are still shedding the virus in their fecal material. And so you're getting a truer picture of the infection in a community, as opposed to testing, which is really, the people who get tested are the people who show symptoms. So you don't really capture the asymptomatic fraction. But again, there are many assumptions to do this, and lots of questions and lots of parameters in the models that we still don't have a good grasp of. The third potential use of this kind of data is with what's called viral evolution. In other words, how does the virus actually change in space and in time as it's circulating around the world or around different communities? So we can compare the genome sequences of the virus, and this is an example of a paper from the UK that shows how the virus has evolved, beginning from its sources in China and the different strains that are circulating around the world. So this is another possible use of this data. A fourth use, which is again probably one of the more useful or important uses, is identifying hotspots. And these are geographic areas that might have higher incidence of COVID 19, which may suggest that public health officials should put more resources into those areas. So I'm showing you a map of Michigan, and it's showing that there are communities, particularly black communities, that are hit harder by the coronavirus, and not just in Detroit, which is the most urban setting in Michigan. And again, this is something that we can direct our resources to, whether it's nursing homes or schools or basically confined spaces where a lot of people are working together. So going back to wastewater surveillance for SARS-CoV-2, again, we are collecting samples from the influent or from the primary solids and then analyzing that. So what are some of the issues? There's a lot of known unknowns. So for example, you are collecting your wastewater quite a distance away from some houses, and there is a possible decay that's happening during this transport time. Sometimes it takes 16 hours, maybe even as long as 24 hours for some of these bigger systems. For the wastewater from your house, you flush your toilet, it goes to the sewer line, and it has to go all the way to the treatment plant, which may be miles and miles away. So how do we figure out that decay that's going on? What about the impact of rainfall, which increases the infiltration and the inflow? Does that dilute the signal? I mean, that makes sense. We still need to do a lot of work to figure this out. What about the impact of temperature? Again, as temperature decreases during the winter or increases over the summer, does that change the behavior or how we quantify the virus? And again, this question of distal versus near contributors. You have a system where you might have houses close to the treatment plant and houses far away from the treatment plant, and figuring out how you can actually account for those differences in distance is again a big unknown. So these are some of the issues. That's why modeling this to quantify the number of infected individuals in a community is still a little bit under question. Sampling also affects the results. Here I'm showing you grab samples, which is basically a one-time dip into the wastewater. And you can see essentially that even if you sample 50 seconds apart from the same waste stream, you're getting this huge variability, as opposed to 24 composite sampling, which is essentially you have a sampler that takes a proportional sample of the flow over 24 hours. Maybe every 15 minutes, it's you have a pump that's taking out the sample, and you can see that the variability decreases a lot, right? You can also see here some of the variability between these two targets, N1 and N2, the red and the green. And even though the general trend is kind of the same, you can see some differences between these two targets. And this is a picture from this paper where basically you can see the composite samples versus the grab samples. And depending on when you do your grab sample, you might be hitting a very, very low point, right? And composite sampling evens out, those points. So something to think about. Another big question is actually data analysis. How do we actually think about normalizing the data? How do we report it? Do we report it per volume of wastewater, per mass of RNA, per mass of solids, per person per day? Or do you normalize to another type of biomarker? For example, this pepper mild mottle virus, PMMOV, has been shown to be correlated to the fecal contribution in the wastewater. So how do you do this? And then how do you relate this to public health data? Is it just the number of confirmed cases? Do you look at COVID-like infections, which is a different number, right? These are people showing up to the hospital with symptoms. Um, they may get tested later or they may not get tested, but it's possible that they have COVID. Do you compare it to other numbers like hospitalizations and so on? So these are a lot of open questions, and people are still working on this as we speak. Then the final piece is, how do you deal with your stakeholders? It's important to be engaged with the different stakeholders, and this includes the community, right? The neighborhoods, the broader public, and of course, you interface with the health departments. And then you have also companies, and here's where we are, researchers. We are also interfacing with the wastewater utilities. So, how do we do this research that basically is community engaged research? And in general, this has been embraced by environmental health researchers, but environmental engineers and scientists still need to learn a lot. Um, Without engagement, we risk not realizing the full public health benefit and also repeating the mistakes of the past. Okay, so now I want to talk a little bit about um, our work as an example. And I'm showing you the Raleigh sewer shed. This is the city of Raleigh, where I live and where North Carolina State University is. And essentially, you can see all the gray. These are all the sewer lines. It's basically showing up as a gray area here. We're also sampling actually from a hospital and also from a couple of neighborhoods. So we call these sub sewer sheds. And the treatment plant is right here, like right there at the bottom, the black dot. We've been doing this since April 10. This particular treatment plant serves 580,000 residents of this county. And we've tested now more than 43 wastewater solid samples. We've tested since April. And we're doing this on the biosolids, the primary solids, not the wastewater. But we also have the wastewater samples, and we're working through those right now. Some initial results are that all solid samples were positive for both N1 and N2. The trends are similar. I'm showing you a graph here, a snapshot where you can see that the N2 and the N1 line up very nicely, on top of each other. In terms of just a detection, as of June 9, just taking an arbitrary date, there were, 78 new cases out of 2,472 cumulative cases. And we can detect it at a 0.22% incidence rate, right? Number of cases per population. So quite sensitive in terms of the technique. Here is actually the latest data from Wake County. And this shows all our data from April to October, end of mid-October. Yeah. And so the top here is the wastewater SARS-CoV-2 data in copies per microgram of RNA. And you can see in April, May, it was still quite low. And then you see the increase in June, right? And basically peaking in July and then also in August. And then slightly decreasing, and then maybe you're seeing something happening there again. And below is the Wake County daily cases. You can kind of see the same shape. Very interesting. So it looks promising. We have not done the statistics on this yet. We still need to actually do some QC on this data, quality control. But it does look like it's correlated, and we'll be fitting models to this data soon, at least for Wake County daily cases. And we also want to use the data to model hospitalizations, COVID-like illnesses, and other health data. And we're waiting for the North Carolina Department of Health and to give us some of the data. So this is the kind of data that you might be seeing from wastewater surveillance. So some of our preliminary results, SARS-CoV-2, N1 and N2 genes are quantifiable both in the wastewater and in the primary solids. Again, we have detected it in every sample that we've tested from April 10. Interestingly, we're finding that the concentration in the solids is much higher than the concentration in the actual wastewater influent, which again is due to the fact that the wastewater solids represents perhaps several days of wastewater. And maybe the virus does partition preferentially to the solids. In the neighborhoods that we have been sampling, we have been detecting them, and also we have been detecting them downstream of that hospital that we were sampling. Also wanted to just show you that we are part of a larger network across the U.S. And even globally. So we are part of this Communicating Sewage Surveillance, or COSIS website, and we were part of as part of this project. We helped develop the National Wastewater Surveillance System that the CDC has now up on its webpage. I've also, been active in talking about this issue in national organizations like the Water Environment Federation. Also given some presentations virtually to groups in the Philippines. And we have also published our results along with, or our thinking at least, the framework with a lot of collaborators in the US and overseas. And in North Carolina, we have been known to be at the forefront of wastewater surveillance for SARS-CoV-2. So, just to summarize some of the critical knowledge gaps, the first is probably method specific, and at this point, I think there is a lot of consensus that the methods do work, and we do have methods that we're quite confident in, and we're working through a lot of our samples right now. Another gap is really figuring out the estimate of the shedding, how much viruses are really shed in each human individual for symptomatic and asymptomatic infections, because we don't know. There's still a huge range, and that affects the variability of our estimates. We don't know about the degradation rates or the decay of the virus, of the genome, actually, the RNA, in the sewer under different conditions. And also just this question of hotspots, using this technique to identify hotspots is still a critical knowledge gap. So to summarize, um, we think that wastewater surveillance for SARS-CoV-2 may be useful, has potential, and primarily for trends tracking. In other words, figuring out, is it increasing in a community? How is it changing over time? Or are our intervention methods effective? Are we seeing the decrease in infections over time? We see this as complementary and not really replacing individual testing, but this gives us a sense of what's going on within a community. And now you can start comparing community versus community, right? City versus city. And you can ask yourself what's working in the city versus another city where you're seeing the infections increase, or the wastewater data shows that the infections are increasing. This kind of work is interesting. It also requires cooperation and partnership. We need public health data from the NC Department of Health. We rely on water resource recovery facilities or wastewater treatment plants for help with sample collection. We know we're accountable to our stakeholders, to the communities that we are sampling from, and it's important to communicate these results to the public also. So our hope is that over time this becomes a method that can be used, um, that would potentially provide public health officials actionable data so they can decide on what to do as we're dealing with this pandemic. And obviously, also as we roll out a vaccine, we can see which communities are becoming more um, covered by the vaccination programs if we see the wastewater data decrease in a particular community. So, with that, I don't know if you have time for questions, but I really appreciate the chance to be in front of all of you today. And good luck with the rest of the convention. Um, and good luck with all the work that you are doing. And congratulations again to the PSBNB. Thank you.